Hello everyone. In the first section of physiology, we'll discuss about blood and its functions. And here is a question before we begin. Take a guess. What is the total length of blood vessels in your body? If all arteries and veins were to join together end to end? Well, the answer is E, 100,000 kilometers. So, just how long is 100,000 kilometers? Well, the circumference of Earth at the equator is 40,000 kilometers. It simply means your blood vessels are long enough to encircle the Earth for 2.5 rounds. Therefore, blood must be very important because we need such an extensive network to transport them in our body. And these are the learning outcomes for this lecture. By the end of this session, you should be able to explain the functions of blood. You should know what are the key constituents of blood, as well as structure and function of hemoglobin, in which you also learn it in biochemistry, and to explain conditions that could arise due to hemoglobin abnormalities. And now, imagine if you could look at all the blood vessels in your body, where could you find them most? Well, the answer is it is in your lungs, as you need many capillaries for gaseous exchange. Then you could also find them in your vital organs, such as your brain, your kidneys, and the liver. And of course, you could also find them around the body. So we need blood to achieve homeostasis, or a stable internal environment. And we have different types of blood cells to perform such functions. First, we will need erythrocytes for gaseous transport. Then we will need platelets to stop bleeding in the case of injury. And we also need leukocytes, or the white blood cells, that fight against pathogen or foreign bodies. Here are the key functions of blood. First, it's needed to transport many different things across the body. Things like gaseous molecules such as oxygen, carbon dioxide, waste products such as urea molecules, hormones, enzymes, nutrients, proteins, and of course the blood cells. Secondly, because blood is mainly composed of water, and water has a very high specific heat capacity, blood then can help to maintain our body temperature at 37 degrees Celsius. Now, on the other hand, blood can also act as natural buffer to maintain our blood pH within a narrow range of 7.35 up to 7.45. Lower than this value of 7.35, you'll have a condition known as acidosis, while higher than 7.45, you will get alkalosis, and in both cases, proteins like enzymes or structural proteins will be denatured. So it's very important to maintain the optimum pH at 7.4. So we can do this because our blood contains the bicarbonate buffer system. On the other hand, we also need blood to remove toxins from our bodies. So toxins will be processed mostly in the liver and in the kidney. As our blood plasma is an aqueous solution, it also regulates the concentration of electrolyte such as potassium and calcium ions, which are then needed for our normal body function. Last but not least, we will need blood for protection. So when injury happens, such as if you get a cut, blood clotting prevents infection and the loss of blood. So if you were to take a blood sample and let it to settle a while by gravity or by centrifugation, it will fractionate automatically into 3 layers. You'll see that the red blood cells are found right at the bottom because they contain iron, so they have a higher specific gravity than other components. While in the middle layer, you will find a very thin layer of yellowish zone which contains the white blood cells and the platelets. And on top, you will find plasma, which is the most abundant component of blood at 55%. So plasma dissolves and transports all kinds of things that I mentioned just now because it is made of mostly water molecules. And now if you look at the plasma, it is basically a complex liquid that many cells are suspended inside, such as erythrocytes or the red blood cells, leukocytes or the white blood cells, thrombocytes or the platelets. And now tell me, which of these cells do not have nuclei? Well, the answer is red blood cells, and the platelets. Both of them do not have any nuclei. Why is it so? Well, first of all, without nucleus, red blood cell can then carry more hemoglobin for oxygen transport. Remember, nucleus is the largest organelle in the cell. While for the platelets, they are required in blood clotting. So they have no nuclei simply because they are derived from the cytoplasm of a mega cell, which is known as the megakaryocyte. So in average, we have about 8% of our total body weight as blood, and that translates into 5 liter for female and 5.5 liter for men, while the maximum blood volume that we can donate is about 400 ml, which is less than 10% of our total blood volume. Again, as I mentioned, if you fractionate the blood sample, you will see 3 layers, which are the top plasma layer, the middle buffy coat layer, which contains both leukocytes and the platelets, and the bottom red blood cells layer. So blood fractionation is a very useful technique in hematology, or the study of blood. And here you have one very important parameter in hematology, which is the hematocrit. Sometimes it's known as the packed cell volume. So hematocrit represents the volume percentage of red blood cells in your blood sample, or simply the portion of red blood cells in your blood sample. So for women, the normal value of hematocrit is about 42%, while for men, it is about 45%. And this explains why men are generally stronger than women, because you get more hemoglobin, and therefore you get a higher aerobic capacity than your counterpart. However, hematocrit is not a constant, and the level of hematocrit can change with the change of the blood plasma level. Remember, hematocrit is calculated as a percentage of red blood cell in the blood sample, and therefore, when the volume changes, the ratio changes as well. And now try this question. What will be the answer? You may pause the video for a while. Well, the answer is increase. As you lose body fluid, plasma level is going to drop. And as a result, the overall percentage of red blood cell is going to increase. So the answer is A, increase. Okay, blood consists of both plasma and the cellular elements. In plasma, 92% is composed of water, while the rest 8% are ions, organic molecules, trace elements, vitamins, and some gaseous molecules. And you have a wide range of organic molecules, such as the following: amino acids, proteins, glucose, lipids, and some waste products. So for proteins, these are the 3 most commonly found blood plasma proteins, which are albumins, globulins, and fibrinogen. For albumins, they are important in maintaining the osmotic pressure in your blood, while for the globulins, these are transport molecules for ions and hormones, and don't forget, they also serve as immunoglobulins, or the antibodies. You also have fibrinogen, which are needed for blood clotting. Then looking at your blood cells, you have the red blood cells or erythrocytes, white blood cells or the leukocytes, platelets or the thrombocytes. And you have 5 different types of leukocytes, like the lymphocytes, monocytes, which is the largest of all. You have neutrophils, eosinophils, and basophils. And your homework now is to look for their basic functions. Again, blood plasma is mostly composed of water. That's because you will need the solvent to solubilize and to transport different materials. As I mentioned, because blood has a very high specific heat capacity, it can hold the heat generated from metabolism and maintain it within a small, narrow range. So for example, when we are overheated, blood will travel to the skin surface for heat dissipation, but when we are cold, blood travels back into the core for heat conservation. So the most abundant organic molecules in the plasma are the plasma proteins. We have discussed them very briefly just now. You have albumin, globulin, and the fibrinogen, while the remaining components are nutrients transported around the body from your big and small intestine, such as glucose, amino acids, fat molecules, and vitamins. Waste products, you have creatinine. It is a breakdown product of the muscle tissue. Bilirubin, a breakdown product of hemoglobin. And urea, a product of protein catabolism. Of course, you do have some dissolved gases, which are basically oxygen and carbon dioxide. So without plasma, you can't transport gases efficiently. And you also need plasma to transport hormones to the site of action. Clinically, it's important to examine the content of blood plasma proteins, and we can do so by running an electrophoresis known as the serum protein electrophoresis, or the SPEP. So a normal electrophoretic pattern will show 5 peaks, which are the albumin, alpha 1, alpha 2, beta, and gamma. Albumin has the highest peak because it is the most abundant plasma protein. Then you have the components of the globulin, which are alpha 1, alpha 2, beta, and gamma. So again, these are the components of globulins. Now my question is, what are the subunits, or where are the subunits of fibrinogen? Well, they are too faint to be seen, so you can't really see them in this SPEP profile. So showing here A and C, these are the normal patterns of the SPEP, so we can use them as references. While in the case of myeloma or a type of blood cancer, you get to see the production of a lot more monoclonal antibodies, which is reflected in the peak of gamma subunits. So this procedure is very cheap and easy, but it reveals a great deal of information. And now let's move on to the cellular components of the blood. And for now, we'll look at the erythrocytes, or the red blood cells. So you should all know that the primary function of red blood cell is for gaseous transport. And to be more specific, it is for the transport of oxygen as well as carbon dioxide. So red blood cells are very pleasing to the eyes because of their color and shape. But this unique shape contributes to the high efficiency of oxygen transport in two ways. First, the biconcave shape of the red blood cell provides a larger surface area, so it can transport more oxygen or carbon dioxide. Second, due to the thinness of the cell, oxygen can then diffuse more rapidly from the exterior to the innermost region of the cell. Now, if you could remember, what is the name of the protein that maintains the biconcave shape of the red blood cell? Well, it is spectrin, and it is a type of cytoskeletal protein. And now let's look at several important facts about the erythrocytes. Number one, they transport oxygen, but they themselves cannot use any of this oxygen that they transport, simply because they do not have mitochondria. Instead, they are undergoing the anaerobic glycolysis, or the lactic acid fermentation, for ATP production. So we have a large amount of red blood cells in our bloodstream, which is about 25 to 30 trillions. But all these red blood cells are having a short lifespan of about 120 days. And that is in comparison to the nerve cells, they can last for our lifetime. But why is it so? That's because all of our red blood cells are constantly exposed to mechanical stress when they pass through very tiny capillaries, inducing wear and tear. So once they become old and damaged, these red blood cells are broken down in the spleen, in which the heme group is converted into bilirubin. So in every second, about 2 to 3 million cells are being made. So that is the very fast speed. And with this speed, you will just need about 8 weeks to replenish all the red blood cells once you donated your blood sample. And therefore, the advice for red blood cell or blood donation is about 2 months, every 2 months. So during its lifespan, a red blood cell will travel about 700 miles or more than 1,000 kilometers. That is the distance equivalent from JB or Johor Bahru all the way to Thailand. And let's look at a bit more details about erythrocytes. Generally, the concentration of red blood cells is higher in males than females. So males are naturally stronger than females, and the volume of red blood cell should be about 80 to 96 femtoliter. So femto is equivalent to 10 to the power of negative 15. So this value is also known as the mean cell volume, the MCV. Lower than 80 femtoliter, you get a condition which is called the microcytic red blood cells. It means that your red blood cells is smaller than usual. And this is usually caused by a deficiency in iron, causing anemia. So I myself is actually showing these signs of microcytic red blood cells from my blood test. and it has been suggested to me that I might have thalassemia. So we'll discuss thalassemia in a minute. So when the MCV is larger than 96 femtoliter, you have a macrocytic red blood cell, which is larger than usual, and that is usually caused by folate or vitamin B9 deficiency. So you might be wondering, since the cells are larger, how can it be anemic? Well, the answer is, although the cells are larger, but their oxygen transport efficiency is compromised. So these cells are larger simply because cell division are delayed, and therefore red blood cell will grow larger before they could divide. And as I've mentioned about the hematocrit just now, there is another important parameter that you should know that we can use to detect blood abnormality. And now, back to basics. What allows the erythrocyte to perform its function? Well, it's all because of the hemoglobin. So hemoglobin is a protein with a quaternary structure because it contains 4 polypeptides, which are the 2 alpha chains and the 2 beta chains. And to be more specific, you have 2 alpha globin subunits and 2 beta globin subunits. And in each of these subunits is having a heme group as the functional group. They can react to oxygen. So in the part 2 of this lecture, I'm going to discuss more on the function of hemoglobin in more details.